Thanks a lot, Wesley, for the introduction. Good morning, everyone. I'm Sutirtha Roy, an MSc student at University of Calgary. I, on behalf of all my co-authors, Moshfik, Jurjan, Richa, and Aditya, will be presenting HoloChemie, a sustainable fabrication approach for creating soft biochemical holographic devices. This biochemical sensing approach adds a new dimension to existing sensing techniques in HCI. For instance, to sense biomarkers in sweat like glucose and lactate for health monitoring and further fitness tracking. Environmental sensing, such as using our CO2 sensors to measure the indoor air quality in a makerspace. pH sensing for both soil and water monitoring, like extremely acidic pH sensing and extremely basic pH sensing. Let's take a look at the existing sensing mechanisms for soft interactive devices. These can be broadly categorized into three types: electrical, mechanical, and electromechanical. While these methods are well explored and provide interesting solutions for soft interactive devices, there's another dimension of sensing that has received far less attention, that is biochemical sensing. This method has the potential to open up new possibilities for sensing in soft devices. On the other hand of the spectrum that contributed chemical sensors and interfaces in HCI, these examples showcase different approaches in this space: UV sensing, pH sensing, and biosensing. However, while these innovations are valuable, they predominantly focus on a single sensing modality, such as either biosensing or environmental sensing. In this work, we, for the first time, explored the use of biomatter to detect chemical changes that can be utilized for various interactive applications. For an interaction perspective, HoloChemie offers several advantages for sensing. Firstly, they allow for batteryless passive sensors by providing colorimetric response. Secondly, they offer wide range of sensing capabilities ranging from wearable biosensing to environmental sensing. Thirdly, to enable real-time quantitative estimation, we contribute a portable hardware system. Finally, to top it all the materials that we used for fabricating these sensors are reusable, biosourced, and biodegradable, contributing a sustainable fabrication approach. However, we faced a set of challenges while fabricating these HoloChemie devices. Existing approaches for fabricating these devices require sophisticated infrastructure such as a wet lab space. We had to design fabrication techniques which are widely accessible and bring these devices outside the clean rooms and wet labs. Existing techniques in physical science research primarily focuses on material formulation with limited emphasis on sustainability. However, here we want to take a complete sustainable approach to ensure that the entire material stack are biosourced and environment friendly. Our contribution to this work are as follows: analyte-sensitive holographic devices for interactive sensing, fabrication approaches for biochemical sensors, comprehensive technical experiments, and interactive application examples for diverse domains. Now, we go into the first contribution. We use the biochemical properties of living matter as the foundation for sensing in our devices. The living matter and biosource materials you see here are the central to the formulation of our biochemical holographic device. Aerococcus viridans bacteria, Aspergillus niger mold, and anthocyanins are these biosource materials. Analyte-sensitive holographic sensors use a functionalized chemical layer on the substrate material that responds to specific analytes like glucose. When the analyte interacts with this layer, it alters the optical properties of the substrate, such as its color, reflectance, or refractive index. Basically, when light is incident on a substrate material, it absorbs light of certain wavelengths. In contact with an assay made up of these biomaterials, there is a change in absorption of light across the UV visible spectrum. To implement this sensing assay, we created custom hardware consisting of LDR and LED arrays for real-time sensing. And here we can see how a user can wear it as a wearable form factor. For fabricating these biochemical sensors, we use different sustainable substrate materials like alginate sheets, non-woven fabrics, chromatography paper, tattoo paper, agarose sheets, and woven fabrics. After preparing these substrates, the next step is to prepare the sensing assays. Glucose detection assay. Here, we have the key chemical components used in the formulation of our glucose sensor: starch, glucose oxidase solution derived from Aspergillus niger mold, potassium iodide solution, and phosphate buffer solution. This assay is prepared by adding a few drops of phosphate buffer saline solution to starch to maintain the conducive pH levels for the enzyme. Then, we add potassium iodide to it. It is then further mixed with glucose oxidase to create the final assay. In this process, the potassium iodide disintegrates into iodide, which further reacts with starch to produce the holographic response. Mix gently to avoid damaging the enzyme. The next is the lactate detection assay. Here we have the key chemical components that we used in the formulation of our lactate sensors. These are starch, lactate oxidase solution derived from Aerococcus viridians bacteria, potassium iodide solution, and phosphate buffer solution. To make it easier for practitioners, we designed the unified approach for these biosensors. In this case, for lactate detection assays, we use lactate oxidase instead of glucose oxidase. Similarly, we add the potassium iodide as mentioned before. Then we add the main component, that is lactate oxidase. Lastly, we again need to mix it carefully to avoid damaging the enzyme. Hibiscus tea assay for extreme pH sensing. We are using commercially available hibiscus tea leaves. We need to boil it for 15 to 20 minutes in water, then either drink it as a tea or just use it as an assay for alkaline pH sensing. Bromothymol blue assay for extreme pH sensing and carbon dioxide sensing. We prepared this solution by dissolving bromothymol blue powder in 0.01 normal of sodium hydroxide solution. Now we will discuss the different fabrication techniques we used to create our biochemical sensors. We employed 3 distinct methods, each offering unique advantages for different applications. So the first one is brushing, spraying, and drop casting. We also conducted a set of technical experiments to validate our devices. Spectrophotometry analysis was performed to measure the spectral response of our sensors. The results show that our sensors have a response in both the visible and UV spectrum. Additionally, we also conducted a user study with 8 healthy participants. The goal was to test the sensor's ability to detect glucose and lactate levels in sweat. The study was carried out under controlled conditions using predefined artificial sweat samples. This approach helped to ensure the consistent biomarker concentrations and enabled us to systematically compare sensor performance across different concentrations of glucose and lactate. We counterbalance the order of presentation of the biomarkers. Like in simple words, each participant had 2 samples, one for lactate and the other for glucose, and then we applied sensors on the forearm near the wrist for 20 to 30 minutes, allowing the participants to continue with their daily activities. Our sensors were successfully able to detect different levels of glucose and lactate in sweat. Skin type of the color did not affect the sensor performance. Participants found the sensors comfortable and unnoticeable during the use. Lastly, now I would like to showcase a range of interactive application examples. Live fatigue prediction in VR exercise. While exercising, a VR display shows the current LDR intensity within the device corresponding to the colorimetric change of the lactate patch. Using our holographic sensor, the user's fatigue point is predicted based on their sweat lactate, and they are reminded to take a break. Sweat glucose monitoring app. We created a mobile app which gave sweat glucose levels in real time. Now that we have seen the contributions, there are a few more things that we need to discuss about these devices. Environmental factors like temperature and pH are critical for enzyme functionality. We used PBS to maintain an ideal pH. The enzymes used in this work function up to 48 degrees Celsius. Since the human skin is 37 degrees Celsius, the sensors work well on the skin without any issues. In this work, we focused on detecting glucose, lactate, CO2, and pH levels. Future research should explore additional biomarkers like stress biomarkers, and additionally, sensing VOCs in makerspace could also be a valuable step for environmental monitoring. For some biochemical reactions, those aren't responses instantaneous, unlike ECG or EMG signals. During our user study, the users wore the sensing patch for like 20 to 30 minutes to assess sensor response and performance. In future, we aim to deploy these sensors in real-world scenarios where participants will wear them for longer durations. So for the conclusion, we developed these biochemical sensors for biological and environmental sensing using eco-friendly materials. Our formulations combining enzymes, plant extracts, and chemicals detect glucose, lactate, pH, and CO2. The holographic sensing technique enables both detection and quantitative measurement of analyte levels. The technical experiments showed precise concentration differentiation, and user studies confirmed glucose and lactate measurement from sweat. We demonstrated different application scenarios and discussed sustainability, limitations, and future directions, aiming to expand biochemical sensing for HCI applications. Thank you, and please let me know if you guys have any questions.
Thank you very much. So we have exactly time for one question, and it would be great if it would be a question from you. Otherwise, so I think it's a super cool project. It's a lot of mixing together chemistry together with HCI and sensor design. That's absolutely fantastic. I was wondering where do you see the future on how we can integrate this into a more digital fabrication approach, right? Will it be something like an automatic textile fabrication machine? Do you see it more in 3D printing? Where do you see the main potential for digital fabrication here?
Yes, that's really a very good question, Michael. I just feel like this idea is really cool to actually create some automatic methods to create out these sensors. So maybe, yes, in future, there are a couple of substrate components like agarose and alginate which we used. Those can be prepared in such a way that it can be further combined with different chemical assays, and by using bioprinters, maybe we can create some patterns out there, and from there we can also, increase out the accuracy of the holograms or holographic sensing out there. So I think that could be a really very good possible case for the future. Thank you.
Okay.